we're doing neurophysiology. And neurophysiology is examining how membrane current is maintained and produced, and how that signaling occurs, and that's what we're going to carry on into muscle. We're going to follow that signal across the neuromuscular junction and how that current is then carried deep down into the muscle, resulting in muscle contraction. And we're supposed to start autonomic nervous system on Tuesday, but I've moved some of the content from the autonomic nervous system to this portion of neurophysiology so that we don't get bogged down while we're doing the autonomic nervous system. So we're going to continue neurophysiology on Tuesday, kind of taking the two topics and overlapping a little bit. But then we'll have autonomic nervous system. An autonomic nervous system is talking about general visceral efferent control and physiology of that as well. So these two sections right here, the basis of neurophysiology and then carrying that into the autonomic nervous system, in my mind, is the major reason why this course is required as a prerequisite for the next semester. Okay? When I took anatomy and physiology, we were actually required to take the second half of the course first. Here you're not allowed to. We were required to as pre-nursing students, because we actually started our nursing program while we were taking the first semester of anatomy and physiology. And we started our cardiac, med-surg, all of that in January. They wanted us to have gone through those systems. So the nursing department asked the biology department to teach the second half of the course first, which, when you look at it, doesn't make any sense, because we were talking about it was a combined course, and we're talking about control of organs, and we haven't learned about the nervous system yet. But the concepts that we cover today and Tuesday in neurophysiology, we're going to start right off with when we talk about the endocrine system first semester of in January of next semester. So if you're taking Bio 431 from me, you'll have already had most of that first lecture. And then we move what we're talking about today with action potentials and depolarization and repolarization, no matter who you take it from. That is the underlying pinnacle of how the heart works. Because the heart doesn't require the nervous system to depolarize. And in fact, I go back and do a review of physiology of how nerves work when I start talking about the heart. And then the autonomic nervous system was what controls reproduction and the heart and digestive system and respiration. So understanding the autonomic nervous system is the underlying basis for the physiology we do next unit, next semester. All right, and then the special senses could have gone in either section. We talked about cranial nerves in our last unit. Obviously, the eye and the ear are innervated by the cranial nerves, but the physiology of how the ear works and the physiology of how the eye works is under autonomic and neurophys. So when I was doing it in the first section, I just kept saying, well, we haven't gotten to this yet, but, and so I decided to finally just move it at the end where those concepts would already be reviewed, and we wouldn't be talking about the physiology aspect itself. So we looked at this diagram of the neuron. When we first started our last unit, just talking about the basic components of a typical motor or multipolar neuron. And today we're going to go back and look at what happens along the dendrite and the membrane of the cell body and how a graded potential is produced. That's a current that can be strong or weak or really strong. That current travels through the cell to the axon hillock. And at the side of the axon hillock triggers a threshold of an action potential. And then we'll see how that action potential goes along the membrane to the synaptic bulb. And that's where we'll stop. Okay? So we should be done between 12:30 and 12:45 with a break in between, because we don't have a lab today. And then we'll pick up on Tuesday with the release of neurotransmitters from the synaptic vesicles and a look at neurotransmitters. Okay? So we could have put those neurotransmitters at the beginning, because this could be, we're just repeating what happened up here, but we're following it with that. There is a lab on the PhysioX lab manual that's assigned for over the weekend. The lab for that is up on D2L. I'll double check and make sure it should be updated because I did that over the summer, but I'll double check today. And I'm going to make it due on Thursday instead of on Tuesday because we won't have completed the synaptic potential part. But you can sure go ahead and we will have done most of it by the time we go today, so you can start on it. If you have more time over the weekend, you're certainly welcome to get started on it. I don't have the packet printed for you, but I'll double check and make sure that the lab up there is the updated one, and it should be, because like I said, I did that over the summer. Okay, so let's look at what a membrane potential is. And this first part, page 2 and 3, if you have the lecture notes, is something new that I put on there. It was a description of how ions travel through protein channels in the membrane, kind of from a layman's perspective, and it gives gives a background. There's nothing in here I'm testing on, but it just gives, for those of you that haven't read it, describes somebody climbing up out of a hole on a ladder as an ion moving through the channel, and relates how the ion is being stripped of its water molecules that are attached to it through ionic bonds and hydrophilic bonds, and they're kind of how it moves its way through and gets extra energy, and it just kind of clarifies what we're talking about the different channels today. Okay, again, it's just more of an understanding rather than a testable information. But why do ions need channels to cross the plasma membrane? So here's our plasma membrane, and the first type of an ion channel we're going to talk about is a leak channel. Old name was leaky, so if I, forget and call it a leaky channel, it's easier to say leaky than leak, and stop and cut it off, so I may slip. But this first illustration up here is of a leak channel. And again, why do ions need channels to cross through a plasma membrane? Back to the early chemistry that I told you to recall from the prereq for the course. What is the chemistry, the biochemistry of the plasma membrane? What are the molecules that make up most of our plasma membrane? Lipids. Phospholipids, primarily lipids with the phosphate on the outside and the lipid tails forming the inside. Remember our lipid bilayer. And what kind of chemical bond connects the carbon-carbon molecules of the fatty acid? Are they sharing the electrons? Are they donating electrons? If they're sharing them, are they shared equally? Do you guys remember any of that? Covalent. So the carbon-carbon bonds are non-polar. The electrons are shared equally between the two carbons. Non-polar covalent. Whereas our ion, our potassium or our sodium or our chloride ion, is an ion. It's got a charge, and that doesn't mix very well with a non-charged molecule. All right, so it's not going to move, even with a channel. It takes, I think that article said, 20 times more energy to, even without an actual potential pump, to move through the channel than, say, a lipid-soluble molecule would to move across the lipid. So potassium ions have a leaky channel, and potassium is in higher concentration inside the cell or outside the cell? Remember? Potassium is inside, and sodium is outside. So we have more potassium ions inside the cell than we do outside. So the drive down the concentration gradient, this is a simple diffusion, is going to be for potassium to move out of the cell. All right, so this is going to be inside the cell, and this is going to be outside the cell. So we also have leak channels for sodium. And sodium is in higher concentration outside the cell. And sodium is going to be driven by its concentration gradient into the cell. So our neurons, and all of this is going to apply to our muscle cells as well. All right, so that's why I put it in this order so we can flow into that concept. Are going to have these leaky channels. However, their numbers are not the same. So in our neuron, as well as in our muscle cell, there are more leak channels for potassium than there are leak channels for sodium. So the nerve cell membrane is said to be more permeable to potassium.
Why are there more for potassium?
I don't know why, they just are.
Okay.
All right, so we can make a cell more permeable by synthesizing more protein and adding more potassium channels, or by doing endocytosis and removing some of the plasma membrane that has potassium channels in it and internalizing it. So we can add or remove these ion channels. That will take some time. It's not going to happen immediately, like opening or closing a gate. But we can affect the permeability in that fashion, all right, by adding or closing these leak channels.
Do they ever share the same channel?
Technically, they can. So many of our ligand-gated channels that we say are, sodium channels or potassium channels, preferentially will allow sodium to leak through, but that doesn't mean that the occasional potassium doesn't. So these are preferentially potassium and sodium, but they're both very small. Typically, they are, I would say never, ever, ever, but typically you would see just the ones that are labeled. Yes?
You said add or remove these channels. The body just creates them and then
Hormones
gets rid of them.
Can change them, can change the behavior.
And is that to
So that would be
change the
this fashion.
concentration?
Any of the channels in here would be internalized.
Okay.
So it can change the behavior of the cell, it can change the charge inside the cell. All right, so those are leak channels. And there's no way of closing them, okay? So the only way we can change the permeability of the cell is to add or to remove them. All right, so we talked about this already, but just
Add or remove the leak channel.
yes. So the ion with the highest concentration outside the cell, sodium ions. Okay, so when we have a leak channel, the drive is going to go into the cell. And when we have a potassium leak channel, the drive is to go out of the cell. Then we have ligand-gated, or if you prefer to say ligand-gated. And these are, ligand is just a word for chemical. So a chemically gated channel. In other words, a molecule has to bind to the outer surface of this channel for it to open. Now, one of the students this morning was talking about how the chemical is removed, and we'll talk about that next week. It depends on the binding affinity. So when it binds, it can immediately be unbound, or it can have a high affinity, and once it's bound, it stays there. Certain cells will make enzymes that will break down any ligands that bind to them, or the neuron that released it may take it back up and break it down. So all of those different processes we will get to, but we'll get to next week. All right, because next week we're talking about the surface of a cell where those neurotransmitters are binding. So that'll be part of that concept, part of that lecture. There are two types of ligand-gated channels. Again, up until last year, I used to talk about this that first week of second semester, the second half of the course. But when we talk about the autonomic nervous system, we talk about this a lot, so I've added this component to this lecture, hence it will be all review and a reminder rather than brand new material. So there are two types of channels that will bind to the neurotransmitter. One is a channel that itself is an ion channel, okay? Maybe I should say there are two types of receptors. One is the receptor is an ion channel and the other receptor is not. And so binding to it has some subsequent steps that have to occur before an ion channel will open. All right? So that first one, the direct opening, is called an ionotropic channel. Or, I'm going to change that, ionotropic receptor. And so it's very similar to a leak channel, except that it has a protein whose shape is determined by the binding of the neurotransmitter. So here the channel is closed, all right, and here a neurotransmitter that binds to this channel. Is in place, and in response to that, the channel is open. Okay, so NT stands for neurotransmitter. So the receptor itself is the channel. Now it may be a channel issue. It may be a channel for potassium or chloride or calcium or sodium ions. It may be for all three and preferentially for one or the other. Okay, it kind of just depends on the protein channel itself. So this is a direct response. It's usually faster, okay, but not as many events have to happen, but there aren't as many of them, and it's usually of a shorter duration. Okay? So you can characterize it as a quick, short, brief response. And that would be the diagram on the left-hand side of the screen. The diagram on the second side of the screen is identified as a metabotropic. From the word metabolism, it sounds, tropic means movement. Sometimes growth. So the metabotropic channel is really, again, not a channel, it's a receptor. And we often see this with hormones.
This is the second one.
This is the second one, okay? So the metabotropic receptor is not going to open. But once a neurotransmitter binds to it, a cascade of events occurs. And I'm just going to give them to you briefly, as you see on this diagram. And then on Tuesday, we'll go into more detail. All right? So when a receptor binds to this channel, let me just get my, when a neurotransmitter binds to the receptor, there is an additional intracellular molecule called a G protein from the guanine amino acid. And that may have already been bound to it, or it will move and bind to it as a result of the neurotransmitter binding. So metabotropic receptors, I want you to associate with the concept of a G protein. The G protein has several components or subunits, and when the receptor binds, one of these subunits dissociates and interacts with another molecule inside a part of the membrane, usually an enzyme. And this enzyme will be activated, and a variety of things can happen, one of which is the opening of an ion channel. the neurotransmitter binding to the receptor did not immediately open the ion channel, but caused a cascade of events which resulted in the ion channel being opened. In addition, other changes inside the cell can occur. The release of calcium, the activation of other enzymes. But the metabotropic receptor is a G protein, and lots of our receptors in the nervous system are this type of receptor. They take a little bit longer to occur, but their actions can be amplified because the release of this subunit can activate hundreds of intracellular molecules known as second messengers. And each of those can open an ion channel. So it's an amplification, okay? So this is short and brief. This takes a little bit longer, but can be expanded, can be amplified. And if that doesn't make sense right now, that's fine. I have a series of images to show you on Tuesday. Yes?
What is the A called?
It's the alpha subunit. It's just the subunit of the G protein. There's alpha, beta, and some other subunits. Always the alpha. It's the alpha that is the one that separates. Yeah. So on Tuesday, when we get through here, all this stuff you're going to learn. So this is what we're going to talk about on Tuesday, and here's the alpha subunit binding to that internal enzyme. So we're just going to go over that and explain that a little bit better on Tuesday. Okay? So I did